microphone, I like to wonder when I talk, so pardon me if I talk into this thing. Anyway, greetings to all of you. My name is Scheibel, Arne Scheibel. I'll be your instructor in Neuroscience 102, which is Functional Neuroanatomy. Introducing, we have, by the way, a wonderful group of teaching assistants, TAs. We have Wenbin, we have Nanthea, Nanthea, there she is. We have Chris Hutchinson. We have a couple of volunteers. We have C. Kim, where's C? There's C. We have Eileen Yu. And we have an archivist, Richard Lamb, who's going to make these things permanent, unfortunately. But there will be, I suppose, some advantage to it, because these, plus an earlier set of tapes, will be available for you down in the Biomet Library if you need review. You're also welcome to take the lectures if you want. But we're here primarily, folks, to enjoy ourselves. I know this is going to be a challenging course for many of you. It has that reputation, and probably realistically so. But it's the most important course you can take. It is your brain. After all, it's what's sending you through UCLA. It's what's sending you through life. When people ask me what did I want to do when I was young, I would say in retrospect what I wanted to do when I was young was either to be a cosmologist or a neuroscientist. And when you think very carefully, there can't be cosmology unless there is a brain to generate this skill. So I think we're money ahead. So I think we have the very best course together that we can possibly have. I'm delighted to see you here. Now, a few words about the course. First of all, and this is important, get to know your TAs and get to know me. I'm going to have 2 sets of office hours each week. The first set I put on the board here, Thursdays, that is a day like today, from 10 till 12, meaning right after the lecture. Then there'll be many of you who can't make that kind of office hour, so I'm going to wait a little bit until your schedules sort of freeze, and I'm going to see when the majority of you can make another cluster of 2 hours. We'll make that my second office hours, plus the fact that I'm always available by email for special groups if you can't get together otherwise. But I do want to get to know you, and I don't want you to have questions flying around that are unanswered. Your questions are very important. Notice on the schedule of lectures that we've told you what we would like you to buy. These things are available, these books are available up at the main bookstore. I think that's most convenient for most of you. First of all, the Human Brain Coloring Book, which a couple of us did some few years ago, and it has stood the test of time. I hate to admit it, but this is used at other schools too, and it is not a Mickey Mouse venture. This is very serious stuff. And the reason behind the coloring is that the kinesthetic input, as you're doing this, as you're making an introductory impersonal diagram, as you're making it yours, you're learning. It becomes a personal document. And then it's very easy to review from. As a matter of fact, and this is true, when I lecture to the residents in neurology or neurosurgery here who are just about ready to go out into the world, you know, and practice medicine, many of them tell me that they've reviewed for their final board examinations using their coloring books, so that it is a kind of thing that should stay with you. By the way, you're free to buy second-hand coloring books as long as they're not colored. If they're colored, that means somebody else has gotten a good out of them, and you need one that's black. The second book we ask you to buy is the Atlas, Neuroanatomy Atlas by Haynes. After the first 2 weeks, you'll certainly live and sleep with the Haynes Neuroanatomy text. It really carries us through the nervous system. Please make sure you have a copy. A used copy is fine, just as long as it's legible to you. The third text is optional. This is The Human Brain by Nolte. A very good, comfortable, readable text for those of you who want a little more information, maybe presented in a slightly different way, but again, following the plan of our lectures, which is very useful. Above all else, your notes are important. In a sense, they are your Bible or your Quran or your other holy book. They're important because they represent what we think. We try to give you the crystallization of what we think is most important in this enormous field of brain. If you have trouble taking notes, do the very best you can. You may want to add a little tape recorder, or you may want to look wistfully at your neighbor's notes or something of that sort. If you have questions, and you all have questions, please jot them down. With those questions, you either ask them here, right in front of everybody, by putting up your hand, and I'll always stop for a legitimate question, or keep the list and come to office hours. I don't want you to run away with unanswered questions. Not that I can answer them all, but the important thing is let's get them out and know where you stand. Let's enjoy it. A couple other things that I know nobody is particularly interested in, but I will mention. By the way, each of you invest another $6 and buy a box of these latex gloves. You'll really need them in the laboratory. We don't want you getting into the formalin and so forth. You know how the course is run. We meet Tuesday and Thursdays sharply at 9. Please come sharply at 9. It's not easy, I know that, traffic and all, but this is a course where missing is rather difficult. My wife teaches this course up at Berkeley, Marion, but she also teaches gross anatomy, and that's a 3-day-a-week course, very, very dense, because histology is included too. And our favorite story is the little Berkeley student who wandered in 4 weeks after the course began and said, I simply have to take your course. I know I'm late, but have I really missed anything? So, yes, they have missed something. So do come right on time, please. What about quizzes? You'll notice that we have 3 quizzes during the 10-week period. It's an awful lot. And then a final. The reason for the quizzes is, well, frankly, it is to keep you on your toes. Folks, there's a little squib at the back of this schedule, and it says something that we all heartfully agree with, and that is, this is a course you cannot cram for. There's a lot of information each time, and each course, each hour, builds on the hour before it. If you try to cram before a course, it's difficult. Most would say it's impossible. So the important thing is keep up with us every day. I suggest that you read the coloring book before you come to lecture. That gives you now remember I said read the first time before you come to lecture. That gives you the general area of what we're going to discuss. You take your notes, and then within the first 12 hours after taking your notes, please reread your notes once. That gives a first reinforcement and starts the process, hopefully, of deposition beginning. Then do color as you begin to study, and the coloring becomes a powerful adjuvant to learning. And you know, I wish this were more like the mathematics or physics course, where with a few facts, you could then reason your way to the answer. Until you have literally all of the hard facts that we're going to give you, it's very hard to reason your way anywhere, because nature is a lousy engineer. She put together things, notice I say she, definitely she. All right, I'll tell you more about that. But she puts together things in a pragmatic way. And you know, pragmatism changes from eon to eon. So if she has a system here that was wonderful for fish, and now the reptile has an entirely different problem to face, she uses the same structure, but sort of messes around a little bit, adds a few tinkles and bells and so forth, and now it has a different shape and form, and yet it's the same system. So we have to be wise enough to see through the schemes, because over about 400 to 500 million years, a rather remarkable system is put together. The greatest biological system that we know of, certainly in the solar system, and for all we know now, perhaps in the whole galaxy. And my effrontery is that I'm asking you to learn in 10 weeks what it took her about 500 million years to put together. So we're going to have to work together on this. It's going to be quite a job. We can do it. In terms of the quizzes, I say 3 quizzes during the year, and a final quiz at the end. How do we mark them? We mark them straight. We don't mark on a curve. As you know, they don't mark on curves at graduate schools. I don't want you competing with each other. I want to see how you best can do, and if you have trouble, we'll get together to ameliorate them to see what we can do to help you. The first 3 quizzes are each worth, I think, 20%. That makes a total of 60. And then the final, which is made up of 2 good quizzes, the final is made up of 40. So 20, 40, 60 for the 3 quizzes, and then the final 40% gives you the mark. We have, I think, the best and also the biggest group of TAs we've ever had in this class. And they will give you more detail about this. But exclusive of this week, which is just starting, they're going to mount a review session every Thursday night. You have a question? Oh, nice to see you. They're going to mount a review session every Thursday night. Is it from 5 to 6 or 4 to 6?
5 to 6 and then 6 to 7 for questions.
All right, 5 or 5:30 to however long it takes. And then before each of the 3 quizzes, they will have a second review quiz on the Friday. So you're going to have very, very good coverage for reviewing. I think this is above and beyond the call of duty, and I'm sure it'll help all of us. I mentioned how I would like you to study. The most important thing, again, is keep up day to day. Do not fall behind. Now, some of you have very ambitious schedules. You're taking a high level of academic points or credits, and many of you are working in the laboratory, which is wonderful. Some of you are doing volunteer work outside. Some of you are showing the United Nations how to do it. You know, you're an active group outside. I'm going to ask you to be very, very thoughtful about how much of this you really want to carry. Those of my students who have had trouble in the past, in part, have had trouble because they were overcommitted. So please reevaluate this very carefully. If you have an advisor, you might want to discuss it with him or her. If you don't, by all means, come to see me or one of the TAs, and we'll try to think with you on this score. I mentioned this before, I mentioned it again. While I talk, while I lecture, if you hear something you don't understand, or if I say something that's wrong, just put your hand up immediately and I'll recognize you. Your questions are usually more important than anything I can say. Now at this point, are there questions about what we're hoping to do and what we plan to do and how you should do it? Yes?
Can you repeat what you were saying about the review session?
Repeat what I said about the what?
The review session?
Yes. There will be each week, exclusive of this one, we're going to this week, exclusive of this one, there will be a review session from about 5 to 6:30 or 7 on Thursday nights. We'll tell you where. And then on in those 3 weeks when we have a quiz, there will be a second review session the following Friday. So in other words, we'll have review sessions both Thursday and Friday of the week where a quiz is scheduled for the following Tuesday, of course. Otherwise, we'll just have a review session on Thursday. And we hope with that, we have awfully good TAs. We hope with that to cover all your questions. Something in back? Yes.
And the one on Fridays at the same time?
And also from 5 to 6:30 or so, yeah.
We
Yeah,
might,
yes.
we might try and do it a little earlier on Friday if that works for everyone, and we'll announce the room numbers on Tuesday.
So Friday may be a little earlier, and the rooms will be announced
On
on
Tuesday.
Tuesday, so you know. I'm hoping we can get the same room all the time, but of
Right.
course that depends on scheduling. Most of the TAs, depending on their own situation, will be scheduling their own times available for you for office hours. They'll let you know. So as I say, make use of the resources. We are here, we really are here to help you. I know there are some courses where the instructor says the main purpose of this course is to fail half of you. Well, my main purpose in this course is not only to have all of you pass, but to pass well and to come out enthralled with the nervous system. It's what makes us human. Matter of fact, let me start now by reading something from a philosopher-scientist named Brunovsky. He said, there are many gifts that are unique in man, but at the center of them all, the root from which all knowledge grows, lies the ability to draw conclusions from what we see to what we do not see, to move our minds through space and time, and to recognize ourselves in the past, on the steps to the present, and in the endless caverns of the future. Our brain sets us free. It lets us wander in space and time. That's the beauty of it. So having said that, let's talk about a little history, long history. Where do we, that is to say, where do these remarkable brains come from? Let's see if this all works. One thing you're going to notice is that the room becomes a little dark. And believe it or not, one of my students from last year said, can't you make that room a little lighter when you lecture? Because I feel I'm always falling asleep. And I'm sure this is as much a comment on the quality of the lecture as it is on the amount of light in the room. But the fact remains, this is about as light as I can make it if you're going to be able to see anything. So I hope you can bear with me and this rather ridiculous way of talking into this machine. Can you all hear me in back?
Yes.
Okay. I want to start by showing you my sexiest picture. It is rather handsome, isn't it? We're looking at the brain, of course, the human brain slightly from behind. Here are the cerebelli. You recognize that. You recognize the great mass of the left hemisphere. And here is the right hemisphere. One of the things you see, of course, is that this brain is furrowed and fissured. It has these little hills, and it has the deep valleys between them. Many of you know that we call these hills gyri, G-Y-R-I. Now, most of these terms should be on the handout sheet, and I do want you to know that. We call these gyri, the little valleys between we call sulci. And we call this kind of brain that is covered with these gyri and sulci, we call it a gyrencephalic brain. The root encephalon, as most of you know, means brain. And this is a brain with gyri, a gyrencephalic brain. Not all brains of mammalian vertebrates, as we're going to see in a moment, are gyrencephalic. Many of them, of the slightly less evolved animals, if any of you have a pet rat or a pet rabbit at home, wonderful little animals, their brains are smooth. We call that a lissencephalic brain, a smooth hemisphered brain. And is there a reason for this? Yes, we think so. One reason at least is that with this fissuring, this kind of thing, you can put 3 pints into a 1-quart container. In other words, you can have more surface area in the same size. I used to go out and teach in the school. I still do sometimes, to little kids, you know, in the 1st and 2nd and 3rd grade. And I point out that the brains of primates and humans, we are just big primates, are gyrencephalic this way, which gives so much more surface area. And I say, would you believe it, that if this brain were smooth, like your pet rats, our brains would be about 3 feet on the side, like this. And I'd say, now what's wrong with that? And one little kid will say, it would be so difficult to get through doorways. And another little kid said, no, the hat sizes would be ridiculous. And then there's always one little kid in back who raises his hand, you know, and he says, getting born. I said, yes, of course, getting born, that's the problem. Because if we had smooth brains and we had to get that big brain through Mama's pelvis, she would have to have a pretty broad pelvis and a ridiculous carrying angle for her femurs. Instead of this way, they'd have to be coming in this way. And it can be shown that physically, this is a very difficult and inopportune way to have to walk. So yes, this is one of those stratagems of nature to make it possible to have more in basically the same space. Now here's a set of brains, and there's a take-home message here. Are you able to see around my pedestal? Notice what we have. Fish, amphibian, reptile, a mammal, in this case a smooth or lissencephalic cortex mammal, a rabbit. Here's a cat who has now, you see, a gyrencephalic brain, and a human. Now the take-home messages here are 2. The first, of course, is the trend from a smooth, naive cortex to a gyrencephalic, ridged cortex. We understand why. The other is, look at the light blue tissue. Blue, right through. This is, as we'll learn, the cerebral or neocortical component. The neocortical component of the brain. And in the fish, it's just a little bit of tissue. And of course it gets progressively larger. And notice how after a while it looms over the whole brain stem and cerebellum. They're all literally covered by this wildly overgrowing blue cerebral hemisphere or neocortex. How did we get a neocortex? I have a feeling that the whole thing is based on smell. The fish were the first vertebrate lines to have active lives, and they lived, of course, in an aqueous medium. And you know, up until then, single-cell organisms and polycellular organisms generally sense the environment by contact. Now, it's very embarrassing to have to know what's up ahead of you by having to bump into it first. If it's something that's edible, well, that's not so bad, you can gobble it up. If it's something that's more likely to eat you, that does become embarrassing. You've waited too long. If it's something sexually interesting, it might be nice to have a little time to get ready for it. As a matter of fact, those are the only 3 eventualities that one faces in the world. Can I eat it? Am I going to be eaten by it? Or can I reproduce with it? Just as easy as that. Nature soon found that if you have floating traces of that distant thing, molecules floating in the media, and if you can sense those molecules, you're way ahead. If you sense molecules that represent something edible, well, obviously you go forward, and in the other case you use other stratagems. Now, there's another part to that. Wouldn't it be nice when you bump into some of these molecules floating toward you from previous experience that you already knew? Did that mean it's edible or it's going to eat me? In other words, it would be nice to carry a trace of the previous experience. That trace we call memory. And my hunch is, even though there are many kinds of memories, as you know, and even single cell animals have a certain primitive kind of reaction that we could call a memory trace, if you will. If we can have a progressive specialization of the front end of the brainstem that keeps traces of previous experience, and then we can compare the sensory input from the molecules we've just sensed, compare that with the trace of previous exposures, we are really money ahead. We do call that a kind of memory. And so certain changes began to occur in brain tissue, and we are beginning just to understand them now. And that, I think, led to the development and the maturation, the perfection of blue tissue like this, which allowed us to hold traces and even to do a bit of, pardon the word, thinking about the meaning of those traces, the genesis of our beautiful cerebral hemispheres. So do remember then this great growth, progressive growth of cerebral cortex and the probable way that it developed. Now in this next picture, which is a cover from Science magazine just a few years ago, it's a whole head section stained by a blue stain called a Nissl stain. Everything was cut through. You see part of the snout here and the eyes and so forth. What's important is this. Do you know what those are? Those are gyri and sulci in the cortex of a mouse. And remember, mice and rats are supposed to be lissencephalic. These folks had discovered a protein molecule which actually triggers gyrencephaly in the mouse. So we have come to the point where we now even know what nature had to synthesize to make the jump from lissencephaly to gyrencephaly. I show you this as an example. I'm not worried that you'll learn anything here, except that, in a sense, the most complex of nature's stratagems will still come back to the genome and to the protein groups. Another sequence that has a take-home message. What do we have here? Rat, ground shrew, tree shrew, tarsius. You all know what a rat is. Ground shrews and tree shrews are little animals called prosimians. They are the earliest introduction to the monkey and primate group. We'll tell you about them in a few minutes. Tarsius is a small monkey. Chimpanzee is a very big monkey indeed. He's an ape. And this, of course, is the biggest ape of all, man. What we have here are areas that are darkened in or hatched or lined or what have you, and a few little areas that are undarkened. See them? Notice that as we go up the scale, the darker areas relatively decrease in size, and the light, unmarked areas increase tremendously in size. Now, what does that mean? Those darker areas are areas of what we call committed cortex. They are primary committed cortex. They are attached either to input systems, bringing in information from below, or they project down output systems, giving direct motor commands to the output. The white area, this represents what we call association or non-committed cortex. And the beauty of that is the association cortex, the non-committed areas, don't get direct information from outside or down below, wherever it is. They get information only from these adjacent committed areas. So in other words, their only job is to take already partly processed information, put it together, and see what comes out. That's the beauty of the primate, and particularly the human, association cortices. This is where we do our higher cognitive activity. It's this kind of thing that made it possible for you all to go to UCLA. Without it, probably would have gone to US. No, I don't. Let's talk very briefly about the development of the human primate brain. This, of course, is a map of Africa. And we know now, it's almost 100% sure, that humanity is of African origin. Running through about 2/3 of the African continent, from down about here in South Africa, right up through here, right up through, Kenya, through parts of Abyssinia, up through the middle of the Red Sea, into the Middle East, and right through Israel and up through Lebanon, is a long rift. It's known as the Great Rift, or Great Rift Basin. It's a depression, and it's really there. You really fall into it unless you stop yourself on the edge. I've been there, and it's quite dramatic. The Great Rift represents what remains, I think, of an enormous slow elevation that developed back, between 150 million years ago. The ground rose, it was all tropical rainforest, and then the ground changed, it apparently began to drop, it became very dry, and it was in this area that the progenitors of mankind developed. Now, let me take you all back to the 60 million year ago period, sometimes known, don't worry about it, as the Cretaceous Tertiary interface. It's when we now have pretty darn good evidence that there was a single or a small cluster of large asteroids that hit the Earth. Many of you know this already. And there was the equivalent, if you will, of a nuclear winter. Most importantly, it was the period when there was a cataclysmic dying off of the most successful animals that nature ever produced. Those, of course, were the dinosaurs. The huge reptiles, they were so successful that some of them were as big as moving trucks. You know, they could be 60, 80 feet long and weigh 30 or 40 tons and very difficult to stand up to. They were just, they ruled the earth. But at the same time, our mammalian ancestors were also present. They were small, they had to be. They lurked. You guys go around lurking? Lurking is a way to keep alive when you're faced with dinosaurs. So they lurked under roots or up in trees and so forth. But when this nuclear winter developed, most of the food supplies were just snuffed out over a period of probably some decades while the remains of the asteroid explosions just swirled around the Earth. And then, with the big beasts gone, the little mammals began to feel their oats, as it were. They were able to come out. They could live more clearly in the trees. They literally took control. So this is the period, around 60 million years ago, when the mammalia, we think, gained ascendancy for the first time. And then, of course, they varied in many, many ways. And this period, this area in here, is going to serve as the developmental ground for a group of mammals that underwent some very interesting changes. Now, first I'm going to show you the skulls of 5 little animals, starting here, going here. And the thing that I want you to notice is 2. The roof line, that is to say, the skull that overlies the brain, gradually arches up as the underlying brain clearly gets bigger. The other thing is that the orbits, the holes where the eyes are, here, gradually move around to anterior, to front. And we're going to find that one of the characteristics of these developing little mammals, as they become pro-simians, that is to say, the beginnings of the monkey group, is that they develop more brain tissue and their eyes go, well, more anteriorly or front. Now what did those animals look like? None of those animals survived. But here are a group of contemporary prosimians that show pretty much the same thing. Some of you have probably heard of lemurs, bush babies, slow loris. There are no fast lorises, by the way. And tarsius. Notice how those little eyes look straight forward at you. Notice also the power of the hind legs, because these had become proud little tree climbers. And as a tree climber, you don't want a paw, you want something you can grip with. Boy, that'll grip. So will those. In other words, paws were becoming hands with little fingers, and the feet the same way. Now, an arboreal existence, we think, fitted us for the kind of life that we later have lived. One of the things, by the way, that characterizes a tree liver is it's nice not to have to go all the way down to the ground and up the next tree. It's nice to be able to jump from tree to tree, right? Very graceful, pragmatic way to do it. However, if you miss, it's embarrassing. So what you want to do is develop, hopefully, some stereoscopic vision. You want to be able to understand spatial distances, just visually. And that's one reason we think why the eyes moved around to a more anterior position, where you had partial overlap of the visual field. And then you had binocular, stereoscopic vision, and you were a much active, much more appropriate jumper, if you will. Now, we think, remember this all started 60 million years ago, we think that maybe halfway through that time, maybe around 30 million years ago or something of that sort, that the nature of this rainforest changed as this swelling subsided and we began to develop a rift. And the ground became drier, the trees were less frequent, and so these animals living in trees had more of a height to go from tree to tree. They no longer could jump from tree to tree, so they had to walk. We think that that was the basis for the development of larger ape-like or primate-like types who, for a number of reasons that we don't understand, began to find it practical to spend part of their walking time in a bipedal position. And you can sort of guess the rest. You go then from increasingly large apes to animals that are even more effective and that may have a little more brain power. Now what I put on the board here, well, with the exception of one misspelling, this of course should be Australopithecus, I'm sorry. Here are some actually rather earlier figures as to the three great, we call them hominid, meaning human-like or man-like, families. Australopithecus, Homo erectus, and Homo sapiens. I have those names down on your handout sheet. Now, in terms of the approximate times when they were living, on this slide, which is now a little out of date, I say that Australopithecines became, there were several actually, more than this, more than Africanus and Robustus, there were several Australopithecines. They became recognizable probably not just 3.5 million years ago, but closer to maybe 5 or even 5.5 million years ago. And they lived probably till about 700,000 years ago. That's a very long run on Broadway. Just think how long that is. You and I, as Homo sapiens sapiens, We're Homo sapiens sapiens. We've been around since maybe 30 or 40,000 years ago. Maybe, give or take another 5. So in a sense, we have so far, a very short run. And unless we're careful, we're going to bring down the curtain quite soon. So we've got to respect these great Australopithecine forebears of ours. What about brain size? You and I have a brain size of anywhere from 1,200 to maybe 1,500 or 1,600 cc's. We're in that area. Australopithecines, around 450 to 500, maybe 550. Their brain size was about a third of ours and just about the size of an adult chimpanzee. So we've had a long way to go. What do they look like? Beautiful skull of an Australopithecine. Not exactly human. Notice there isn't that much frontal lobe here, but there are tremendous bony ridges, because this fellow must have had tremendous muscles for chewing. His eyes look straight forward, but look at those powerful supraorbital ridges. We're, of course, interested in the brain that was in here, and we can imagine a 450, maybe a 500 cc brain in here. Let's put meat on his bones. What would he look like? Whoops, sorry. Here's what he would look like. Isn't that touching? Isn't that powerful? Obviously not human. Obviously you wouldn't expect to see this individual walking in the halls of UCLA. Nevertheless, there is the feeling of understanding, of beginning to be able to put things. I find this a very moving theoretical reproduction of what one of our ancestors maybe 2 or 3 million years ago might have looked like. And here is a group of them living in the world they knew, a family group, Papa, Mama, Junior. And the site for all this was Africa, because we think of the rather peculiar geographic and meteorological changes that occurred at this period. Now here are just some actual skulls. These are the various kinds of Australopithecines. Don't worry about it. But they all, you see, are characterized by a relatively small skull, large eyebrows, and so forth. Now probably around 1 million years ago, I wasn't there, at least I was born shortly thereafter, but we had the first great migration out of Africa. And we don't know what all of the triggering things were. It might have been, again, meteorological, but an individual with a little more brain size, known as Homo erectus, migrated out of Africa, and in the succeeding tens of thousands of years, probably got up into Europe, but certainly got as far as mainland China and the South Seas. There were many land bridges, of course, at that time. Homo erectus, with a brain size of probably about 1,000 cc's, was already a much more intellectually capable organism than Australopithecus. Homo erectus, we believe, discovered how to use fire. And that when he discovered how to use fire, he could not only keep himself warm, he could, well, to be charitable, he could cook food, or we could say he could burn food. But the interesting thing is that when you cook or burn food, you cut down the amount of digestion time that is necessary in your own stomach. And with that, less digestion time, you're less tired after a good meal of meat. You have time to lie around the cave mouth and gossip. And many people feel that Homo erectus was the first gossiper because he had time and perhaps was developing laryngeal changes that might support some primitive language. Here are the brains of the skulls of several other of these forms. Homo erectus, notice looking a lot more human, more of a skull case for brain. Now, following Homo erectus, also we think, coming out of Africa, although that is not so clear anymore, was the third great mass of hominid humanity. We call it Homo sapiens. We seem to be the latest derivative of Homo sapiens, and we therefore call Homo sapiens sapiens, which you've all heard of Neanderthal man. And Homo sapiens neanderthalensis was apparently the first of our clan, if you will, to catch a hold in Europe, move into the Middle East, probably farther. And the exciting thing is that Homo neanderthalensis, appearing perhaps, a couple 100,000 years ago, almost certainly, the evidence now is clear, almost certainly overlapped with the latest immigrant, again from Africa, our forebears, Homo sapiens sapiens. And so here we have this intriguing picture of temporal overlap between a more primitive Homo sapiens, that is the Neanderthal clan, and ourselves, sapiens sapiens, overlapping for anywhere from 10 to 40,000 years. And the big question is, do any of us have Neanderthal genes? A few USC students suggest to me that they do. But no, that's not fair. But the data are getting increasingly clear that there is very little genetic evidence that there was any significant sexual interaction between these two. It seems difficult to believe, but it may be true. So perhaps the only trace that the Neanderthal man leaves in our culture are those cultural or historic memories of giants in the woods or monsters living under the culvert. Some of you who have read Hans Christian Andersen have read some of these things. It's only a guess. Maybe Bigfoot or Quisquash, whatever they call these men of the hills that apparently have vanished recently. Perhaps they were the very last gasp of Homo neanderthalensis. Maybe. Here's a presumed Neanderthal family. Notice that they are imagined as dark in color and still perhaps rather hairy. This is not necessarily true because Neanderthal, we know, wandered as far as northern Europe, and there every bit of sunlight would be important. And perhaps they look more like this. Give these people a shave and you would never notice them if they walked right down the corridor next to you. So you see, we're really rubbing elbows with our forebears, and I find it very exciting. And by the way, in case we get a little too huffy about our brain size, I told you that our brain size varies from 1200 to 1500 cc, and that Homo erectus, the first hominid form that migrated out of Africa, averaged about 1,000. We have some, in history, we have some measurements of the brains of so-called great people. Now, Dean Swift, who wrote Gulliver's Travels, had a very big brain. His brain was 2,000 cc's. Anatole France, the great French novelist, lived only about 125 years ago. Anatole France's brain size was 1,000 cc's. So he had no more brain than a Homo erectus, and look what he did with it. So the brain is a very flexible instrument, and size alone is not a measure of capability. But nevertheless, there probably is some kind of, what should we say, some kind of magic dividing line. Maybe it's around 800 or 900 cc, where certain kinds of cognitive functions first become possible. Just the last thing I want to show you is, here are scrapers, artifacts that people produced while they were alive. The people are gone, the artifact is still here. Primitive scrapers from 1,700,000 years ago. A less primitive scraper from 500,000 years ago. There is 1,200,000 years between this group and this group. Technology did not advance very quickly then, obviously. But look at this. beautiful, beautiful spearhead. This was done about 17,000 years ago. The jump in technology as we get to the brain of the Homo sapiens sapiens is obvious. A beautiful female figure, also 17 to 20,000 years ago, found in the Czechoslovakian area. Magnificent. Faceless as they often were. What do they emphasize? They emphasize what's important at that time. Breasts and hips, right? That's where children come from. And in terms of finish and conception, this could be a beautiful, modern, impressionistic sculpture. All is not always good. Along with all the good human drives come bad human drives. And here is an old Australopithecine skull with a depressed fracture. This poor guy apparently was hit in the head by a friend. Well, he was a friend. Apparently some kind of battle axe. And look what he did. Look at this. This is the breastbone, a sternum, pierced by a stone arrow. It went deep enough probably to injure the heart and kill this person. So you see there's aggression at every level, and of course if I finish off with this, this is Hiroshima. So we know that humankind brings with it cognitive delight, humanity, beauty, and also aggression. And these are the things that we have to wrestle with. So folks, that's sort of the beginning of it all. Make sure you get your books and your gloves, and we'll meet bright and early on Tuesday morning.